The bacterial transformation process or the experiment was first conducted by a British medical officer, Frederick Griffith, in 1928. Frederick Griffith proved that a particular material that is transferred from one bacteria to another bacteria to convert the second bacteria into first one. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about bacterial transformation process, also known as DNA as the genetic material. So this was the first experiment conducted by Frederick Griffith in 1928. He then worked with Diplococcus pneumoniae. So he worked on Diplococcus pneumoniae. So some books have provided that Griffith worked on Diplococcus pneumoniae, and some books provided that Griffith was experiment on Streptococcus pneumoniae. So in this case we are taking the Diplococcus pneumoniae for example. Okay, so Diplococcus pneumoniae is basically a bacteria that causing the pneumonia. Okay, so Diplococcus pneumoniae, when Griffith worked on this Diplococcus pneumoniae, he observed the two variant of this Diplococcus pneumoniae. One variant is known as R variant and another variant is known as S variant. So what is mean by R and S? So R variant means for rough and S variant stands for smooth. That means these R variant are avirulent and S variants are virulent. Okay, so whenever a bacteria is virulent, that bacteria creates a disease in our body, and whenever a bacteria is avirulent, this bacteria does not create any disease in our body because this kind of bacteria are digested by our system. But S variants or smooth variants are does not digested by our system, so that they can cause serious illness in our body. So basically, what is the difference between two variants? So whenever we consider about R variant, so it is the rough variant. So this is the structure of Diplococcus pneumoniae. Okay, so whenever this variant is naked variant and its surface is rough, so it is called as or known as rough variant. But in the case of smooth variant, the surface is rough also, but there is a presence of polysaccharide coating in outer surface of this particular bacteria. So this stands for smooth or S. Okay, so in the case of smooth variant, there is a polysaccharide coating present. So here a polysaccharide coating present on the outer surface of this Diplococcus pneumoniae. So these Diplococcus pneumoniae are does not digested by our system and they causes serious illness to our body. And in the meantime, Frederick Griffith also discovered three strains of this particular Diplococcus pneumoniae: the first, second and third strain, and the two variant of this strain, so 1R and 1S, 2R and 2S, 3R and 3S. And it is observed that a specific genetic mutation is responsible for creating those variants. Okay, so in a gene there is a mutation. By this mutation, this R variant can shift into S variant. Okay, in a particular mutation, this R variant can shift in S variant, and in other mutation, that is the double mutation, the S variant can shift with R variant. Okay, that means whenever a mutation occurs, then there is polysaccharide coating occurs. That means the gene are activated for the polysaccharide coating, and those particular genes are actually basically made a polysaccharide coating on the outer surface of these bacteria, and whenever there is no mutation occur, then these R surface or rough surface is present. So these rough surface are easily digested by our gastric juices, and so they are known as avirulent strains or avirulent varieties. But in this case of virulent varieties, they are does not digested by our system, so they cause a serious illness to our body. So what is the experiment of Frederick Griffith that was conducted in 1928? So before I start that, Frederick Griffith chooses two different variant of the different strains. That means choice Griffith's choice of variants. Okay, so Griffith choose 2R and 3S for his experiment. Why Griffith choose 2R and 3S? Because there is a theory that concludes that if there is a mutation occurs, 2R can be converted into 2S. Okay, so if a mutation is occurred, 2R can converted into 2S, and also 3R is converted with 3S is converted with 3R. Okay, so this is the form of reverse mutation or double mutation. So whenever it cannot be concluded that this 2R or 3S have there some specific characters or the genetic materials that has been transferred from one bacteria to another. So Griffith conducted 4 different experiments. So in first experiment, Griffith chose 2R live bacteria. So 2R live bacteria, and then Griffith injected this 2R live variant into a live mice. So inject this 2R live variant into mice, and after few hours or few days, Griffith observed that these mice is survived or lived live condition. And when Griffith obtained the serum from this particular mice. So whenever Griffith obtained the serum from these mice, there are no bacteria variant. So no bacteria variant are present in these mice. Okay, so this is the first experiment. In the second experiment, Griffith chose 3S live variant. Whenever Griffith injected this live variant or 3S live variant into mice, after few time or few days, Griffith observed that these mice are dead. So this is the reason of death of these bacteria. And whenever Griffith collected the serum from these dead mice, Griffith isolates a 3S variant from the serum of these particular mice. And in the third experiment, Griffith took 3S variants, but in this case it is heat killed. Okay, it is heat killed. When injected into a mice, after few days, Griffith observed the mice is live, and whenever he collected the serum from these mice, there is also no bacteria present or absence of any kind of bacteria strains or variants. Okay, so this is the third experiment, and the fourth experiment is the most crucial experiment for Griffith. So in the fourth experiment, Griffith chose 2R live bacteria along with 3S variants, that is heat killed. So what is mean? That means Griffith applied heat to kill the 3s variants, and along with 2R life, both are combined and makes a cocktail of bacteria or Diplococcus pneumoniae and injected into mice. So whenever he injected this cocktail into a mice variant or mice model, so injecting, after few days Griffith observed this mice is dead. So this mice is dead, and whenever Griffith collected the serum of this particular variant of serum of this particular mice, Griffith observed there is a presence of 3s live bacteria, 3s live variant. Firstly, Griffith injected 2R live along with 3s heat killed, but in the resultants Griffith observed 3s live bacteria. Okay, so there is a 3s live variant, but when Griffith injected, these 3s are heat killed. So Griffith concluded that there is some substance present. This substance comes from 3s to 2R and converts these 2R bacteria or 2R variants into 3s pathogenic bacteria. Okay, so convert it into 3s variant. So some materials that comes from 3s to 2R and convert this particular 2R variant in 3s pathogenic variant. So this is the reason of the death of the mice. So this is the conclusion of Griffith, that some materials comes from 3s to 2R, and this particular material convert this 2R variant into 3s pathogenic variant, and this is resultants of the death of the mice. So why Griffith choose 2R and 3s variants? So in case here, as I told you earlier, that whenever this 2R or 2s is present in there, so R can be transformed to S and S can be transformed to R by a single mutation or genetic mutation. So it not been possible to conclude that some genetic material comes from S variant to R and convert it to S variant. Okay, whenever Griffith choose these two different strains 2R to 3A and 3S, it is now been easy to conclude that the sum of the genetic material or some materials comes from 3S to 2R and convert it into 3S pathogenic variant. So this is why Griffith choose the two different variant or two different strains to conclude his experiment, and it is clearly defines that two variants are different and single genetic mutation doesn't cause 2R to 3S mutation or 2S to 3S conversion or 3S to 2R conversion. So there is some substance present, so these substances can be traveled from 3S to 2R and convert the 2R variant into 3S pathogenic variant. I hope this video is helpful for you to understand the classical experiment of Griffith's or the bacterial transformation experiment. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.